Welcome back to OncoTrial Insights with Dr. Pidugu, the podcast where we dive into the latest breakthroughs in clinical trials and cancer research and how they impact patient care. Today, we're excited to have Dr. Pidugu with us to discuss an intriguing study known as the LUMINOSITY Trial. Dr. Pidugu, thank you so much for being here.
Thank you, Christina. It's great to be here and chatting with our listeners. Just a quick heads up before we get started. The views expressed on this channel are solely my own and do not represent those of any institution, organization, or employer. This content is for educational and informational purposes only. Please consult your healthcare provider before making any healthcare decision. The information I will discuss is not meant to endorse or criticize any specific products or brands. My goal is to provide you with educational content, shedding light on various topics.
That's an important disclaimer, Dr. Pidugu. Now, let's dive into some exciting advancements in the treatment of non-small cell lung cancer, NSCLC. A recent study published in the Journal of Clinical Oncology highlights the use of telisotuzumab vedotin in patients with previously treated c-MET protein, overexpressing advanced nonsquamous EGFR-wildtype non-small cell lung cancer. For those in our audience who may not be as familiar, could you briefly explain what non-small cell lung cancer is and why innovations in its treatment are so critical?
Certainly. In the United States, lung cancer projections for 2025 estimate approximately 226,650 new cases, 110,680 in men, and 115,970 in women, with around 124,730 expected deaths, 64,190 in men, and 60,540 in women. Non-small cell lung cancer, NSCLC, accounts for about 85% of all lung cancer cases. Originating from the lung's epithelial cells, NSCLC generally progresses more slowly than small cell lung cancer, SCLC. NSCLC is further classified into subtypes based on cellular morphology, including adenocarcinoma, squamous cell carcinoma, and large cell carcinoma. Diagnosis typically involves imaging, CT and PET scans, followed by a biopsy. Molecular testing is often conducted to detect genetic alterations such as EGFR, ALK, KRAS, or MET mutations, which guide treatment decisions. Prognosis in non-small cell lung cancer varies by disease stage and molecular profile. While early stage NSCLC may be effectively treated with surgery or localized therapy, advanced stage disease remains challenging. Within NSCLC, non-squamous subtypes, including adenocarcinoma and large cell carcinoma, form a significant group. Among these, EGFR-wildtype NSCLC is often treated with chemotherapy, followed by immunotherapy or targeted therapies based on patient-specific characteristics. A key molecular feature in this subgroup is c-MET protein overexpression, present in about 25% of EGFR wild-type non-squamous NSCLC cases, although reported prevalence ranges from 13.7% to 63.7%. This overexpression is associated with poor outcomes, such as a reduced overall and progression-free survival, and is linked to resistance to EGFR targeted therapies. c-MET pathway activation may result from overexpression, gene mutations, amplifications, or exon 14 skipping mutations. These insights have led to the development of targeted treatments, including MET inhibitors and antibody-drug conjugates, ADCs. One such promising ADC is telisotuzumab vedotin, designed for tumors with high c-MET expression, which is typically measured using immunohistochemistry. Overall, c-MET overexpression plays a critical role in disease progression and treatment resistance in EGFR wild-type non-squamous NSCLC and represents an important target for personalized therapeutic strategies.
So, Dr. Pidugu, the name Luminosity certainly evokes a sense of hope. Could you start by telling our listeners what this trial was all about?
Absolutely. The LUMINOSITY Trial was a phase 2 study that focused on a specific type of lung cancer called non-small cell lung cancer, or NSCLC. Specifically, it looked at patients whose cancer had progressed after previous treatments and whose tumors showed an overexpression of a protein called c-MET. The trial investigated a new therapy called telisotuzumab vedotin, or Teliso-V for short.
Teliso-V, that sounds like a targeted therapy. Can you explain how it works?
Exactly. Teliso-V is what we call an antibody-drug conjugate ADC. It's designed to specifically target cancer cells that have a high amount of the c-Met protein on their surface. The antibody part of Teliso-V acts like a guided missile, seeking out and binding to these c-Met proteins. Once it binds, the cell internalizes the drug, which then releases a potent anti-cancer agent, ultimately leading to the death of the cancer cell.
That's a very precise approach. The trial focused on a specific subgroup of NSCLC patients, those with nonsquamous EGFR-wildtype tumors exhibiting c-Met overexpression. Why this particular group?
That's a crucial point. Previous research suggested that patients with this specific combination of characteristics might be more likely to respond to c-Met targeted therapies. The LUMINOSITY trial aimed to confirm this and see how effective Teliso-V could be in this population where treatment options can be limited after initial therapies fail.
Can you please walk us through the LUMINOSITY trial design and the patient population that was studied?
Sure. LUMINOSITY was a phase 2 multicenter non-randomized two-stage study. In stage 1, patients were enrolled into 3 cohorts based on tumor histology and EGFR mutation status. Cohort 1, c-Met protein overexpressing, nonsquamous EGFR wildtype NSCLC. Cohort 2, c-Met protein overexpressing, nonsquamous EGFR mutant NSCLC. Cohort 3, c-Met protein overexpressing, squamous NSCLC. Based on predefined expansion criteria from stage 1, stage 2 proceeded with enrollment of patients from the group that met those criteria. As a result, stage 2 included patients with c-Met protein overexpressing, nonsquamous EGFR wildtype NSCLC.
And what were the key findings of the LUMINOSITY trial in this selected group of patients?
The results were encouraging. Teliso-V demonstrated what we call durable responses, meaning the treatment continued to be effective for a significant period in some patients. Overall response rate of 28.6% in the nonsquamous EGFR wild-type NSCLC cohort, and this rate was notably higher, 34.6%, in patients who had particularly high levels of c-Met expression. Most patients experienced a reduction in tumor size. The median time to response, as assessed by Independent Central Review, ICR, was 1.41 months across all patients with c-Met overexpression. The median duration of response, DOR, was 9.0 months for c-Met high, 7.2 months for c-Met intermediate, 8.3 months for the overall c-Met overexpression group. The proportion of responders maintaining a response for 6 months or longer was 63.0% for c-Met high, 47.4% for c-Met intermediate, 56.5% overall. The median progression-free survival, PFS, was 5.5 months for c-Met high, 6.0 months for c-Met intermediate, 5.7 months overall. The median follow-up duration was 20.2 months for c-Met high, 18.9 months for c-Met intermediate, 19.3 months overall. The median overall survival, OS, was 14.6 months for c-Met high, 14.2 months for c-Met intermediate, 14.5 months overall. This suggests that the level of c-Met on the tumor cells could be a predictive factor for how well a patient might respond to Teliso-V.
That's a significant finding. What about the safety profile of Teliso-V? Were there any notable side effects?
Like all cancer treatments, Teliso-V does have potential side effects. In the LUMINOSITY trial, the most common treatment-related adverse events in the nonsquamous EGFR wild-type NSCLC group were peripheral sensory neuropathy, which is nerve damage causing tingling or numbness, peripheral edema or swelling, and fatigue. Most of these were manageable. However, some serious adverse events, notably interstitial lung disease, ILD, in some patients. Peripheral neuropathy was also a common reason for treatment discontinuation, though these events often occurred in patients who were on treatment for an extended period.
The mention of serious adverse events like interstitial lung disease is concerning. How does this balance with the promising response rates seen in the trial?
That's a critical consideration in oncology. While the response rates, especially in the high c-MET expressing group, are exciting and offer hope for a difficult to treat patient population, the safety profile needs careful evaluation. The occurrence of interstitial lung disease highlights the importance of close monitoring and understanding which patients might be at higher risk. The researchers are undoubtedly analyzing these events to better understand and potentially mitigate these risks in future studies and clinical practice.
Were there any limitations to this phase 2 study that we should be aware of?
Yes, that's an important point to address. The primary limitation of the phase 2 LUMINOSITY trial is that it did not have a direct comparator arm. This means that all patients in the study received Teliso-V, and there was no group receiving a placebo or another active treatment for direct comparison within the trial itself. While the study demonstrated encouraging responses, especially in the high c-MET expressing group, the lack of a control group makes it challenging to definitively say how much better Teliso-V is compared to the natural course of the disease or other potential treatments in this specific patient population. The researchers did compare the results to historical data from studies involving standard of care treatments like docetaxel. However, because c-MET overexpression is often associated with worse outcomes in NSCLC, directly comparing these historical results to the LUMINOSITY patient group may not be entirely accurate.
That's an important distinction. It sounds like the ongoing phase 3 trial is designed to address this limitation.
Exactly. Based on the promising results from the phase 2 LUMINOSITY trial, especially in the c-MET protein overexpressing nonsquamous EGFR-wildtype NSCLC population, an ongoing global randomized phase 3 study called TeliMET NSCLC-01 is currently comparing Teliso-V monotherapy directly to docetaxel in this specific patient population. This randomized controlled design will provide the more robust evidence needed to determine the true efficacy and safety profile of Teliso-V compared to the current standard of care.
This is certainly an exciting development for patients with this type of lung cancer. Dr. Pidugu, considering the limitations of the phase 2 study, what is the overall significance of its findings?
Despite the lack of a comparator arm in the phase 2 trial, the LUMINOSITY results are still significant. They provide compelling evidence that Teliso-V can induce durable responses in a specific subset of pretreated NSCLC patients who have c-MET overexpression, particularly those with high levels of this protein. The identification of c-MET expression level as a potential predictive biomarker is also a valuable contribution. While the safety profile, especially the risk of interstitial lung disease, needs careful monitoring, these phase 2 findings strongly warrant the ongoing phase 3 trial. The LUMINOSITY trial has successfully identified a patient population that may benefit from this targeted therapy and has paved the way for a definitive study that could potentially lead to a new treatment option.
Overall, how do you see these results shaping clinical practice moving forward?
The LUMINOSITY trial indicates that telisotuzumab vedotin Teliso-V could become a valuable treatment option for patients with previously treated c-MET overexpressing EGFR-wildtype NSCLC. While safety concerns like interstitial lung disease require careful monitoring, the durable responses seen, particularly in patients with high c-MET expression, support the potential for more personalized targeted therapy. These findings have already led to the FDA granting accelerated approval for Teliso-V on May 14, 2025 in this patient population. I'm excited to look forward to the phase 3 trial results, which will provide more definitive data to guide clinical practice.
Dr. Pidugu, thank you so much for shedding light on the LUMINOSITY trial, its findings, and its limitations. Your expertise is greatly appreciated.
Thank you, Christina. It's vital for patients and their families to stay informed about these kinds of advancements.
That wraps up our conversation with Dr. Pidugu on the groundbreaking Phase 2 LUMINOSITY trial and what it means for patients with advanced NSCLC with high c-MET protein overexpression. Thanks for joining us on OncoTrial Insights with Dr. Pidugu. If you enjoyed this episode, don't forget to subscribe, leave a review, and share it with your colleagues or anyone who might find it helpful. We'll be back soon with more updates on the latest clinical trials and advances in cancer care. Until then, stay curious, stay informed, and keep pushing for better outcomes.